hand over to Liz. And there we go. We are go. Thanks ever so much for agreeing to this, and we're looking forward to what you've got. If people could want to ask questions, feel free to chat, put them in the chat when you think of them. We'll take the questions all at the end. And as you've just seen, we are recording this session, so people can log on whatever time zone they're in at any different point to come back to it. Thank you very much, Liz. Really looking forward to your talk.
Great, Tim. Yeah, thank you so much for both for the invitation to speak as well as the nice introduction. As Tim pointed out, we've known each other for quite a while now, right? So it's kind of it's nice to sort of see where Tim has gone since I first met him as a postdoc. So the seminar I want to give today is, I'm going to tell you about two, sort of the biology we're interested in. But what I was trying to overview, sort of based on my initial conversations with Tim and Rob about this, was sort of how we're using FCS as a tool to look at disordered proteins and their interaction partners. And so I'm going to sort of give an example of the ways we're applying this method. And so I'm happily to answer questions both about sort of the applications as well as the biology behind it. So I'd start off by thanking my lab, so the people that do this work. And so this is the current lab, but a lot of what I will tell you about is actually built upon the shoulders of former lab members who are listed here. And of course, various funding agencies for us being able to do this. Okay, so my lab is interested in intrinsically disordered proteins. So, as a very cartoon view of globular proteins, let me do this. All right. Of globular proteins, we can represent those as sort of this very simple circle here. And for those of you who are maybe students, you probably have primarily learned about globular proteins when you've studied protein biochemistry, right? So these are proteins with well-defined tertiary structures that have hallmarks of classical elements of secondary structure, alpha helices and beta sheets, and they form these very compact structures. So just for contrast, and I know this is probably reviewed to a lot of you, intrinsically disordered proteins are kind of at the opposite end of the spectrum here, and it's represented in this cartoon form by this wet spaghetti noodle looking thing. And that's because they do not form stable secondary and tertiary structures in general. And instead they have sequences that are defined by their lack of those hydrophobic sequences that drive the collapse that is necessary for globular proteins, but instead are enriched and charged sequences and highly polar sequences, which allows them to remain unstructured in solution. And really, this is sort of a spectrum in between something that's fully globular and something that's fully disordered, and that you have many, many proteins, probably the vast majority of proteins that are somewhere in between, and that they may have well-folded domains that are then linked by these relatively long unstructured linkers or flanked by these relatively long unstructured linkers. And so typically when we talk about disordered proteins or disordered regions, we're defining things that are, something on the order of 30 residues or longer. So, short things like loops that are also very dominant in protein structures are typically not described as disordered regions. So because their sequences are so different from globular proteins, and because they don't have these well-defined sort of structural interfaces in globular proteins, they often interact with their binding partners through relatively small, short sequences, sometimes defined as SLIMs or morphs, or there's a bunch of acronyms that can differentiate between various flavors of these that are located in the disordered regions. And so you may have multiple of these within a specific region. You may have different SLIMs or different morphs for different binding partners for the same disordered region. And often these interactions are dominated by charge, right? So charge-charge interactions are the most common kind you'll see within disordered regions. So when we think about how disordered proteins or regions bind their partners, there's also a whole variety of flavors in the way they interact. And so this top one may be the one that is sort of the most familiar to people with a structural biology or biochemistry background, and that is the classical folding upon binding. So in this case, the disorder is really a consequence of being isolated in solution, but upon interacting with a binding partner, it may become partially or entirely folded. That's what we see here. But then, in the past few years or decade now, I guess, right, since we began to use tools like NMR, which were really good at, and also computational simulations, which were good at being able to map what disordered proteins look like upon binding to globular partners. We also know that they don't necessarily, or it's not required that they become structured when they bind to their partners, and that they may bind in one relatively well-defined but still unstructured conformation, or they may form these relatively dynamic complexes where you have a number of different types of possible interactions between the disordered protein and its globular interface. So the point of the slide is just sort of to highlight the complexity of the disordered protein systems, both studying them in isolation, but as well as starting to characterize them when they interact with their partners. And because these are often not amenable to structural techniques that are really good at large things like cryo-EM, we don't necessarily get a lot of information from these methods, then we use other biophysical techniques, and single molecule fluorescence being one of the ones that I think has proven to be most powerful in terms of what insight it has provided to us upon both sort of the conformational ensembles that are sampled by disordered proteins, both in solution as well as when interacting with binding partners, but then also quantifying the binding interactions themselves. And that's kind of one of the things I'm gonna focus on in this talk. So the two proteins that we study in my lab predominantly are both entirely disordered, and we study them because it provides us a nice way to be able to contrast their similarities and differences, sort of with the ultimate goal of being able to, provide sort of general insight into functional interactions and of these disordered proteins. So the first part of my story is going to focus on the protein tau. Tau was first identified and described as a stabilizer of microtubule dynamics, so it binds to microtubules in cells. But maybe 10 years after it was first identified as having a role in microtubule stability, it was also identified as the primary component of the neurofibrillary tangles that are the hallmark of Alzheimer's disease and other tauopathies. So as a consequence, I would say there's probably been a lot more work on looking at tau aggregation. So basically what happens in disease as opposed to, tau function, although there's certainly been some very nice work on that as well. These are two very different types of behavior, right? So this is a functional interaction between tau and the globular microtubules, or even the microtubule lattice. And here you have tau interacting with itself in order to form aggregates. The other protein we study is alpha-synuclein. So like tau, this is a neuronal protein. It has a much more poorly defined function. We don't really know what it does in cells. It probably binds to membranes, and it probably has some role in synaptic vesicle trafficking regulation, but that has not really been defined on a molecular or mechanistic level. But like tau in disease, it also self-aggregates and is the primary pathological fibrillar aggregate in Parkinson's disease and other synucleopathies. So they have these things in common, but they are otherwise very different proteins from the perspective of thinking of them from the disordered protein level. And so we are able to really contrast their behaviors. So I'd say the question that really motivates the research in my lab is, or the talk I want to get today is like, how can we characterize binding interactions and functional mechanisms of disordered proteins? And this is sort of given what we know about them and that they may not become structured when they bind, that they may bind transiently or weakly. And so what are the best tools we can use to try and understand more about these binding interactions? And in tau, I'm going to focus primarily on the functional interactions with microtubules. All right, so let's start with tau. And so this is just what I've already told you about tau. And then there's just images. Oh, and I forgot a citation for that one. I'm sorry. But this is on the disease part. So this is a neurofibrillary tangle. So you can see in this brain slice, you can see that this is a stained, this dark area are the neurofibrillary tangles. These are intracellular deposits, primarily of tau, that you find in Alzheimer's disease. And this is from a more recent work from the Kinney lab, where you can see tau is the green dotted along the microtubules, which are in red, and you can sort of see in this particular conditions, it's forming these sort of like little clusters along the surface of the microtubule. Okay, so let's walk through the domains of tau that we know about, and so overall it's a pretty big protein for something that's entirely disordered. And I will say there actually is a cryo-EM structure from the Nogales lab that came out a few years ago. And the sort of really striking thing about that structure, they were able to identify this central region here, this microtubule binding region, which, as the name suggests, is thought to be the primary tubulin or microtubule binding site for tau. And they were able to show that it basically binds along the surface of the microtubule, but it doesn't become structured in in doing so. So this is a nice example of a disordered protein that, at least upon binding to tubulin or microtubules, does not appear to really take on canonical structure. So we have this central microtubule binding region, which is about 130 residues. It is flanked by the proline-rich region, which are 2 proline-rich regions, which are about the 100 residues upstream of that. And then it has the N-terminal domain, and then the short C-terminus. So there are 6 isoforms of tau that are expressed in the adult brain. And those isoforms are formed by splicing, alternative splicing of this R2. So one of these microtubule binding repeats gets eliminated, and either one or both of these N-terminal inserts. And so that's what makes up the 6 different isoforms. Interestingly enough, in the developing brain, you only get the shortest isoform of tau. So it's missing both of these inserts as well as this R2 insert. And so that's one of the questions we're exploring, is whether or not you have functional differences in the way that isoform of tau is regulated versus the isoforms that dominate in the adult brain. So these R regions are called repeat regions, and that's because they're relatively poorly conserved, but somewhat conserved sequences that modulate the interaction with the microtubules. And then this R prime region is also one that's become of interest both to us and others lately. Doesn't share as much sequence similarity as these other repeats, but it's highly conserved among species. And in our hands, at least, it certainly seems to have a strong effect on interactions with soluble tubulin. Okay, so the first work I want to show you is we're focusing on this fragment of tau that I'm showing you here. So you can see that it contains one of the proline-rich domains, all 4 of the conserved microtubule binding repeats, and then this R prime domain. And this is a fragment of tau that is commonly used for biophysical studies, and there's 2 reasons for that. One is that the sequences that drive aggregation of tau are located in R2 and R3. So from an aggregation perspective, this short fragment of tau recapitulates some of the behavior of the full-length tau. And it aggregates much, much better. So when you're talking about getting experiments to work, then that's a reason people often use fragments, is because they just behave better in the lab. Interestingly, this proline-rich region, it's been known for years that you need to include at least part of the proline-rich region in order to get significant binding to tubulin and microtubules. And so this is a functional form of tau. If you add this fragment to tubulin, it's capable of polymerizing the tubulin into microtubules. Okay, and I guess I should give just at least a brief overview of FCS in case there are no people who are not familiar with the method, even though I imagine a lot of you are, so just for contrast here. So like many single molecule techniques or diffusion-based single molecule techniques, at the basis of this method is a highly focused laser beam, which I'm showing in cartoon form here. So you end up with an excitation volume here that's on the order of femtoliters. Like a lot of these methods too, you're allowing Brownian motion to be your primary driver of changes in signal. So we let our molecules just diffuse through as they normally would due to thermal fluctuations. And we quantify the fluctuations in our signal intensity through a temporal autocorrelation, which gives rise to the curves that look like this. And the decay rate of this curve tell you something about the underlying temporal dynamics that give rise to these fluctuations. And so how we traditionally use FCS in my lab, and how many others do too, is you take many curves and you average these curves together. So this is what is shown down here, is these gray curves are many individual curves that are overlying each other. And then the average curve is shown in purple. You can see it has a lot less noise. And then you use the magnitude of the fluctuations as a weighting factor for fitting these curves to a variety of models. And this is something that allows you to weigh less heavily areas where you have more noise. So in many cases, if you're trying to measure accurate diffusion times or diffusion coefficients, it's a more rigorous way to analyze your data. And that's what we have done in my lab for years and years. But then I want to show you some data that my student Xiaohan collected. Well, when he was my student, he's now a postdoc. And so this is looking at tau in the absence of tubulin and in the presence of tubulin. And so this is just soluble tubulin. We have tau with a fluorescent label on it, and we've added unlabeled tubulin to it. And so in the absence of tubulin, you can see that we have these individual gray curves. You can't make out the individual curves, but what you can see is they all kind of sit on top of each other because our average red curve is sitting right in the middle of them. But in the presence of tubulin, he noticed that, on an average curve looks still, more or less in the middle of these, but we can see there's a lot more spread in terms of the individual autocorrelation curves. And so the thing that Xiaohan thought was that we were, by averaging all this data and looking at the average of the curves, we were maybe losing a lot of the information that might actually be useful in learning about how tau interacts with soluble tubulin. So rather than doing this averaging I talked about, he decided he would instead fit the individual curves and get distributions of diffusion times and try to use those as a way of understanding this interaction. All right, so this is then a plot of what this data looks like. So on the y-axis, I have our diffusion times, and on the x-axis, I have two different tubulin binding proteins. So in both cases, the tubulin is not labeled and the binding protein is labeled. So this P1234R prime is my tau construct. And this protein DARPin is this little engineered protein that binds to tubulin with a 1-to-1 stoichiometry. And so we've analyzed the data the same in both ways. And what you'll notice right away is, first of all, not only is the average tau tubulin complex have a larger diffusion time, so it's much bigger than the average tau DARPin complex, but the distribution is much wider. So, this data, we have it all clustered very narrowly around 0.6 in our milliseconds in our diffusion time, whereas we have something like 0.65 to about 1.4 milliseconds in our diffusion time here. So this tells you right away that the type of interaction between our tau construct and tubulin is very different. It's certainly not binding with 1-to-1 stoichiometry like the DARPin is. So our conclusion from here is that we're getting some sort of heterogeneous assemblies. And I will note that this is, even though we're mixing tau and tubulin together, we don't have GTP present. So we're not seeing polymerization into microtubules, but simply the binding of these tau constructs to our tubulin dimers. All right, so what Xiaohan was interested in doing was understanding how the various repeats impacted sort of the size of these assemblies that we're forming. So we have two color-coded sets of data here. This pink data is any construct that has all 4 of the microtubule binding repeats. And then this red data has 3 of the 4 microtubule binding repeats. So just to illustrate here, P123 means it has P2, R1, R2, and R3. And so what you'll notice is we have what we would call both natural constructs, meaning the constructs are in the order you would find them biologically, so like P1234, and then we have some where he has basically made cloning constructs where he's switched, for example, the position of R2 and R3. And so there are 2 reasons that we're doing this. One, we're interested in how many of these repeats, how the number of repeats impact binding, but then also the order of repeats, like is there specificity in terms of this interaction? And so what you can see for these is that for anything that has 4 binding repeats, you can see that they all form distributions that have on average about the same size and the same width in terms of their distribution. And on average, anything that has 4 repeats is larger than anything that has 3 repeats, right? But then when we get down to the 3 repeats, you can see that actually the repeat matters, right? So when we have something that has both 2 and 3, even if they're in opposite order, these on average form larger assemblies than those that are missing either 2 or 3, right? So it's kind of an interesting, what our conclusion from this is that as long as the repeat is there, it doesn't really matter what order the repeat is in, but some repeats have a higher affinity than other repeats, right? So for example, we can contrast these 2 and we can see that the combination of 3-2 leads to larger assemblies than something with 3-4. And so then we might conclude from this data as a whole is that repeats 2 and 3 appear to be particularly important in terms of forming larger size assemblies. And so this could mean that the binding sites within these domains have a slightly higher affinity, and so they hold on to tubulin complexes a little bit longer. Because what we're thinking when we have a distribution like this is we have at any given moment, we have somewhere between 1 and 4 tubulin bound here, right? So that's why we have this wide distribution. Something down here maybe has 1 tubulin bound, whereas something up here may have 4 tubulins bound. And this is what this orange line actually represents. So this orange line is what we would expect for these constructs. So the lower line is for a construct that has 3 repeats, and the upper line is a construct that has 4 repeats. That's what we'd expect for a 1-to-1 complex. So that's just sort of giving you a ballpark. Over here, we can see that a lot of what we're getting seems to be consistent with, 1 or 2 tubulin bound, right? Whereas up here, almost everything has more than 1 tubulin bound, right? It has multiple tubulin bound. Okay, so this again, this is kind of, I guess this, it still blows my mind a little bit, right? Like, we typically think, the first thing you learn when you learn about protein structure, right, is that sequence dictates structure, dictates function, right? And so what we're showing here is we can basically move these sequences around and still get something that appears to be relatively functional in terms of the interactions it has with tubulin. And functional too, in that all of these guys will actually polymerize tubulin into microtubules. So now what we've done is we've taken this diffusion time data, which I had on the previous slide, and just taken the average value of that diffusion time, the median diffusion time, and we're plotting that for each construct against its rate of tubulin polymerization, right? So just to orient you, these are the big complexes down here, so the larger diffusion times, and these are the fast polymerizers up here, so the high K observed, right? So this construct, our longest construct, which contains everything, is also our fastest polymerizer. Okay, so if we look at the data here, we can see there's a pretty nice linear correlation between something that binds more tubulin and polymerizes tubulin more rapidly. And so maybe that is intuitively, when we think about it, that makes sense, because in order tubulin polymerization is a nucleation dependent event, meaning it requires a certain number of tubulin to come in contact with each other in order for microtubules to grow. And so by increasing the number of tubulin dimers we have associated with tau, we're basically increasing the probability that that nucleus forms and that polymerization occurs. Right. And so the other thing you'll see is that these scrambled constructs, these constructs which do not occur biologically, are perfectly good at polymerizing tubulin, right? So P1234 is a natural construct. And you can see its polymerization rate is really, really similar to P1324, which is not a natural construct. We switch these two guys around. And so not only do they bind tubulin, they seem perfectly capable of polymerizing microtubules. And what you look at this little cluster down here, so again, but these two orange lines. So these are the orange lines I had previously, which sort of show you the one-to-one complexes. And you can see these that are the poor tubulin binders are also those that are going to polymerize tubulin most poorly into microtubules. They're just not as good at it because they're not able to bind as much tubulin along a single tau construct. Okay. So full-length tau back up here again. So, sort of as our interests evolved, one of the things that we recognized is that there's been and kind of, as I already said, there's been a lot of attention played to this microtubule binding region for the very reasons I said, that it's core aggregation domain as well as thought to be the primary binding site for microtubules. And so much less attention has been given to this proline-rich region in the N-terminal domain. And so where that sort of led us was asking the question is, what roles do these domains outside the MTBR play in tau function? And specifically, to the next few slides, I'm going to tell you about focus on the proline-rich region and the N-terminal domain. Okay, so there's tau again. Now we have our new construct, which has both parts of the proline-rich region here. So in contrast to the previous one. And so now we're using the same type of FCS analysis, and we're looking at binding of this construct to tubulin. And what you can see, probably don't remember the diffusion times I had earlier, but this one forms even larger and sort of a wider spread of diffusion times than the previous one. And it's able to form relatively large tau tubulin complexes. But what's interesting is if we include this N-terminal domain, and so these are named about their absence or presence of those N-terminal inserts. So for example, tau 0N is missing both N1 and N2, whereas tau 2N has both of them. And this tau 1N star is actually what we would call our sort of biophysical construct, in that it doesn't exist naturally. So that's just N2. But the natural one has only N1, but in this 1N star, we've eliminated N1 and we have only N2. So what you'll see, interestingly enough, is that all of these inhibit the binding of tubulin, right? So when the N-terminus is present, we on average have complexes that are smaller in their average size, but that all of them are pretty good at doing it. So you can see this distribution of tau 2N is by far sort of narrowest distribution, but all of them are significantly smaller than this construct that is lacking any of this N-terminal region. And so we see that the N-terminus is a negative regulator in full-length tau, and it reduces sort of the size of these heterogeneous tau tubulin complexes. Again, this is sort of an interesting feature of disordered proteins. In this particular case, so this is a student, Kristen, who did this work. She took the sequence of N1 and N2 and she completely scrambled it. So all the same amino acids, but just randomly scrambled, and she made a construct that we call tau in scrambled. And what you can see is that is basically having that completely scrambled sequence kind of has the same negative regulation of binding that the wild type protein does. So again, this is maybe a step up from what Xiaohan was doing, sort of swapping out domains, but showing that even the sequence itself is not so important there. I would say, with the caveat that she did keep all the amino acids the same, so it had the same overall charge and hydrophobicity, and so maybe that's important. But as long as we have those residues there somewhere, it's pretty good in terms of being able to regulate the interaction with tubulin. And as it decreases binding, it also inhibits the polymerization. So these are now the actual polymerization curves where we mix tau and tubulin and GTP, and then as a function of time at 37 degrees, we can see an increase in scatter, which allows us to see that microtubules are forming. And what you can see is this PRR MTBR prime construct polymerizes more rapidly than any of the other constructs. And then this tau 2N, which kind of shuts down our binding most effectively, is also our slowest polymerizer. Okay. So given that we know that this construct lacking the N-terminus really binds, forms very large tau tubulin complexes, and we'd already looked at the role of the different repeats in binding, that's what Xiaohan had done, Kristen was interested in understanding what the proline-rich region on its own was doing. So she made a construct which was just the proline-rich region, which is shown here. And then using FCS, she looked at tubulin binding, and so she saw something pretty remarkable. So in this case, we're looking at a full binding curve. So she's increasing the concentration of tubulin along the x-axis, and we're measuring these diffusion time distributions along the y-axis. And so if we start with this proline-rich data, which is shown in orange, you can see that these distributions, even as a function of tubulin, remain relatively narrow, right? You can also see that we see an increase in the diffusion time as tubulin binds. And you can see that that saturates at around maybe 2 or 2.5 micromolar tubulin for this particular condition. If we then go to this longer construct, we can see that at low concentrations, so up to 1 micromolar tubulin, these data overlay each other pretty well, right? We have narrow distributions, the average diffusion times, which are normalized to the size of the tau constructs, I would say, are more or less the same. And it's only when we exceed this concentration of around 2 micromolar that all of a sudden we begin to get these really large complexes that are forming with this construct that do not form with this construct, right? So how we have interpreted this data is that this proline-rich region actually serves as the primary tight binding site for tubulin. And the reason we think this is true is, first of all, we know that it can bind because we wouldn't see this increase in diffusion time for the PR alone if it didn't bind. But the fact that these data overlay each other so well at these low concentrations suggests to us that even in the context of this longer construct, we're populating the binding sites in P1 and P2 first. And it's only after we've populated those binding sites do we begin to fill in binding sites along this microtubule binding region. And not only that, these are relatively weak transient binding sites, whereas this looks like a relatively tight stoichiometric binding, right? And so I'm going to show you on the next slide, I'm going to show you this data again, but in a different way, right? So this is taking, again, the PRR, and that's in orange. That's the same data I just showed you, but now we've averaged all those points together and we've plotted them on a different axis on the y-axis. So you can see really nicely that that looks like a nice cooperative binding curve. And what you can really see there too is that there's kind of a break here at about 1 micromolar. That's really consistent with this data. And so what we think is that you have this one tight binding site where you have 1 tubulin binding, and then you have a second binding site where the second tubulin is binding, and that it's binding very nicely and stoichiometrically. We know that this from some earlier work that Xiaohan did, that this diffusion time here is consistent with 1 PRR bound to 2 tubulin dimers, right? But for contrast, I want you to look at the other constructs we're looking at. So if we look at the gray, this is a construct where we have only the microtubule binding region. So then what's really striking about this is you can see that despite its name, in the absence of the proline-rich region, it really doesn't like to bind soluble tubulin. And if we include this R prime as we have in yellow, we can see that we get a little bit of binding, but you can also see the shape of this curve, which mostly looks like sort of a weak non-specific interaction, right? We don't see the strong cooperativity we see with the proline-rich region on its own, which is what we see here. Right, so from this, we know that this PRR is capable of binding tubulin, but then what was really beautiful is that she showed it's also capable of independent polymerization. So this is this light scattering assay I talked about earlier, where we have all the ingredients together for actually polymerization to occur, and with the same color coding, so orange is the PRR, you can see that's actually the only one of these constructs that's capable of polymerizing. And we did the EM just to show that we're actually making microtubules, because tau can form all sorts of crazy structures with tubulin. But in this case, you can see we're actually making classical microtubules with just the PRR, right? And so all of this, so all this is sort of consistent with our idea that, okay, the PRR has a really critical role in binding to microtubules, directly binding to tubulin and microtubules. It's been described as a regulator in the past, but in our view, based on what we've seen, it's probably a primary binder, and that the microtubule binding region has a separate tubulin binding role. So certainly it accelerates polymerization. I showed you that with Xiaohan's earlier data, but it's not the primary binding site. And just like the full-length constructs, if we take these proline-rich region and add the N-terminus back to it, we can see that it diminishes the binding. So again, even in the absence of the microtubule binding domain, so this is FCS data again, sorry, this is diffusion time along here as a function of tubulin concentration, you can see that this nice cooperative curve now becomes much, much weaker when this N-terminus is here and we have the same negative regulation of binding. Okay, so this sort of gets to a little bit of the biology that we're working on now, which is understanding a little bit more about the proline-rich region. So that this stretch of the PRR is the region of tau that is most heavily modified by phosphorylation as opposed to translational modification. And many of you, even if you're not working with tau yourself, if you've heard anything about it, one of the things you may have heard in Alzheimer's disease is that it's hyperphosphorylated, right? And so what this just means is it has a higher abundance of phosphates on it than it normally does, although I would say we don't have a great idea about what normal phosphorylation of tau looks like. But within this proline-rich region, there are about 30 possible phosphorylation sites that have been identified in both healthy brain and postmortem Alzheimer's tissue. So we know that they're there. And so because this is in our hands as a primary tubulin binding site, it would make sense then that phosphorylation is a major regulator of how tau interacts with microtubules. And of course, we have several theories, hypotheses about how this works, but one of the things we're exploring is like, can this do this by direct modulation of the binding, right? So this is a new set of data, but this is from another student, Karen, more recent. And you can see this is the PRR alone binding a tubulin as measured by FCS. And you can see her curves like remarkably similar to the curves that Kristen generated. But she started working with phosphomimic. So she took 4 of these sites within the proline-rich region that have been most commonly identified as being abnormally phosphorylated in Alzheimer's disease. And she wanted to see how these interfere or whether they interfere with tubulin binding. And so this first cluster of data in blue, she has a single phosphomimic at each of these sites. And you can see that it does diminish binding, not significantly, and it seems to plateau out here about the same place as wild type, but there's maybe a slight diminishment of binding. It binds a little bit more weakly in the sort of mid-ranges of tubulin. If she puts 2 of these in combination, you can see that we begin to knock down that binding a little bit more, right? And then if she has all 4 of them there, then we have really significantly diminished binding, right? So we're able to really knock down the interaction. And so what we're trying to do now is understand how this phosphorylation may also, which is basically changing the charge distribution in this PRR, may also impact how this N-terminus that I've told you regulates binding, how phosphorylation may impact how the N-terminus is regulating the binding for the proline-rich region. That's just, sorry, all the data as a whole, so you can see all of it there. Okay, so that wraps up what I wanted to tell you about tau. So that was, sort of the ways we are using FCS as a means of understanding how tau interacts with this soluble partner. And one of the things we're doing right now, actually in collaboration with a colleague of mine who's a chemical biologist, is we've been able to use a semisynthesis technique to introduce bona fide phosphorylation at sites 231 and 235 within tau. And so we're very hopeful that we will soon be able to determine whether or not a phosphomimic that we have here actually recapitulates the same effects as bona fide phosphorylation. And if not, what effect the bona fide phosphorylation has on this interaction. So hopefully that's coming soon. And I want to switch gears then in the last, I don't know, 10, 15 minutes, tell you a little bit about how we were using FCS to look at alpha-synuclein. So this is the slide, this text, sorry, is what I already showed you with alpha-synuclein about why we care about it. This is a stained slice with a Lewy body in it. So that's the intracellular inclusion that's found in, extracellular inclusion that's found in Parkinson's disease. And then this is my schematic of alpha-synuclein. So it's smaller than tau. It's only 140 amino acids. It has this sort of the predominant domain is this 100 residue membrane binding domain. And unlike tau, the membrane binding of alpha-synuclein becomes helical when it binds to membranes. So it seems that it forms an amphipathic helix, and then it binds along the surface of the lipid bilayer. So it's not a transmembrane domain, but it binds along the surface, where it has an abundance of lysine residues within this N-terminus that probably mediate favorable interactions with negative phospholipids that are found in the lipid bilayer. This NAC is named as a non-amyloid component. So this is first found actually in Alzheimer's disease patients as co-aggregated with amyloid beta, but this is sort of the central aggregation prone region of alpha-synuclein. And then it has this really highly charged acidic C-terminal. So by highly charged, I mean it has a charge of -13 spread over 40 residues. So it's very negatively charged, and it's been proposed to interact with a lot of different partners. I think physiological significance still has to be established for a lot of those. Within the membrane binding domain, you have a number of residues, mutants, sorry, that are point mutations that are implicated in early onset Alzheimer's disease. Parkinson's disease. So that's a pretty strong link between the role of alpha-synuclein and disease, as not only is it the primary component of these plaques, but you have point mutations that lead to early onset disease. So like tau, synuclein is also heavily modified by post-translational modifications. So this is a relatively recent review where it just is showing you all the different residues that have been identified as possibly being modified, and these are the possible modifications. But I want to draw your attention to actually one that is not on here, and that is the N-terminal acetylation of synuclein. And the reason I want to highlight that is, well, not only is because we're interested in it, of course, but because it's really different from all of these side chain modifications. So side chain modifications such as lysine acetylation or phosphorylation, as you probably know, are regulatory modifications, right? There are enzymes that put on that modification, and there are enzymes that take off that modification. And as we saw in the case of tau, that may change how it interacts with a binding partner, right? Or somehow change its downstream fate after, say, a phosphate group is put on or removed. N-terminal acetylation, unlike these others, is a constitutive modification. So what I mean by that is that it's thought that N-terminal acetylation happens co-translationally. So the N-terminal acetylase actually associates with the ribosome and acetylates the nascent chain as it's coming off the ribosome. But there's also no known N-terminal deacetylases, meaning we don't have an enzymatic way to deacetylate the N-terminus. So for the majority of proteins, that means that once it's on there, it's on there. So you can think of this as the physiological form of the protein. Troponin is one of the best studied examples of sort of a constitutively internally acetylated protein where it's known to be critical for its interaction with actin. So it binds to actin much, much better when it is acetylated. And that is the reason for the vast majority of years that people have studied, tropomyosin, not troponin, have studied tropomyosin is because they have to extract it from animal sources, because bacterial enzymes don't typically put the post-translational modifications you need your proteins to have, your mammalian proteins to have. They lack the machinery to do this. And so in that case, you often are purifying it from animal sources or doing other things to get the protein. So my lab was quite lucky in that around the time we became interested in N-terminal acetylation, the Mulvihill group was interested in N-terminal acetylation of tropomyosin, and they created a construct where you could co-express NATB complex, so the N-terminal acetylase enzyme complex from yeast with in a bacterial system, and it would acetylate your protein during synthesis, and you could purify internally acetylated protein. And so we got the plasmid for this from them, and we found that it worked beautifully with alpha-synuclein. So we could co-express this, and we would end up with synuclein that was bearing this N-terminal acetylase. And we were very excited. And then we did a whole bunch of disappointing biophysics with it because it didn't seem to make much of a difference, right? So, us and others found that it had relatively mild effects on how alpha-synuclein behaved, which was, contrary to what I'd expected and somewhat disappointing. Okay, and I'll say that was until my postdoc Melissa came along and showed that it actually has a major impact. And so what I'm showing you now these are primary mouse neurons. And what she has done is she's growing these in culture, and then she is treating them with aggregates of alpha-synuclein and allowing them to incubate. So this is a mechanism. This is a exploration of toxic mechanisms of alpha-synuclein, whereby the idea that has become popular in the field, and there's some good evidence for it, is that you may have, these amyloid diseases often spread in a very characteristic path through the brain, meaning it initiates in a single region of the brain and then spreads to other regions in a very characteristic manner. And the idea is that you basically have transmission from the initial stage of aggregate formation to other regions through release of aggregates that are then internalized by the neighboring neurons. And so this is how it's able to propagate through the brain, is by this sort of cycle of, you take up some aggregate, you aggregate all the endogenous protein within the cell dies, it releases the aggregate, and then that can be internalized by other neighboring neurons. And so it propagates through the brain this way. And so in this model system, what we're trying to do is look at internalization of these alpha-synuclein aggregates by these primary neurons. And so this first day 7, this is day 7 post-culture, where she has, day 7 post the time where we added synuclein to our non-control cells. And what we're staining for here in blue is just tubulin, so you can see where the cell outline is. And then in red, we're staining for this phosphoserine 129 alpha-synuclein. So what this phosphoserine 129 synuclein is, it's just a modification on synuclein that is upregulated. You find more of it present as the synuclein begins to aggregate. So it's our way to look at our amount of synuclein aggregate. So you can see we have a little bit in the cell body under sort of normal conditions. But for cells that we've treated with alpha-synuclein aggregates, that's what the PFF is. You can see that by day 3, we're seeing a whole lot of that red signal already. And then by day 7, it's pretty much filled up the cell body as well as spread down to the neurites. And I'll tell you that by, we only measure through day 7 because by day 8, so many of our neurons are dead. We can't really, take pretty images of them, right? So what's striking is if I contrast this treatment with fibers made with the acetylated protein with those made from protein that's lacking that acetylation. So you can see that if we compare day 3, these look barely more so than our control cells. By day 7, you're seeing a stronger pathology where you see the staining of the phosphoserine 129 in the cell body. And these will live longer. So we can take data at day 10 and you see the eventual spreading, but it's not nearly, I would say, as dramatic as what we see for the acetylated protein, right? So this was sort of our first result where we saw a major difference in how acetylated proteins seem to interact with some potentially physiological partner. And that's through being a much more sort of virulent infector of neurons and much more capable of causing the aggregation of the endogenous protein. And I will say mechanistically, we still don't know exactly why. We're exploring this, right? You can imagine that these acetylated protein maybe is internalized more rapidly. That's one of the things we think is true, but we're still working on that. You could also imagine that the acetylated protein is better at seeding the endogenous protein. And that's because the endogenous protein is also bearing that acetyl group and maybe it just seeds better. That's also possible. We don't know mechanistically, so right now this is sort of phenomenological, like we know that this is true, but mechanistically we're still trying to figure out why it's true. All right, so then this is our attempts to try to learn more about how it's interacting with the cell, right? So, sort of we can think, okay, it gets internalized by the cell, but what's the pathway? Who's it interacting with? What's it binding on the cell? Like, so let's think about cellular partners that might be important for this internalization. And there's certainly been a lot of work on alpha-synuclein and cell membranes, including work done in my lab many years ago. This is where this model from the Denise lab shows this sort of long alpha helix of alpha-synuclein binding along a planar bilayer. But a lot of this work has been done on pure lipid bilayers, and we know that cell membranes are a whole lot more complex. And in particular, we're adding protein exogenously, right? So the extracellular side. So not only is it seeing the membrane, it's seeing all these proteins and all these glycans. So, cell membranes are heavily glycosylated. And so we thought, okay, let's use cell membranes that look, membranes that look a little bit more like cell membranes and see what we can learn from that. And so Melissa made these giant plasma membrane vesicles. So she used chemical treatment, and you get these cells to basically give off these giant plasma membrane blebs, which contain all the plasma membrane components. And they're microns in size, even though that has no scale bar. And so that's nice because they're optically resolvable. So we can image them, right? And so to do this, we then put a fluorescent tag on our alpha-synuclein. And we first made these GPMVs with SHSY5Y cells. And what you can see is that we can see these nice outlines, not very smooth, not like this picture here, but not, we can see these outlines of the alpha-synuclein binding to these SHSY5Y cells. Interestingly enough, if we made GPMVs with hex cells instead of SHSY5Y cells, the only reason we really see these GPMVs at all is because there's so much background from the synuclein that doesn't bind, right? So very clearly we can contrast how these two bind. And so we were trying to think, okay, there's clearly differences in whatever things are found in the cell membranes between these two that drive this interaction. So one of the things that seemed straightforward for us to consider were the glycans that are found on these cells. So we did try a variety of endoglycosidases to cut off different types of sugars. And what we found is that if we treated these with an enzyme called PNGase F, which cleaves off end-linked glycans right here at the first glycosidic bond, then we actually reduced the binding of synuclein to these SHSY5Y cell GPMVs. And to quantify this, we used FCS. So now we're coming back to the FCS, right? So again, this is using it in a little bit of a different way than FCS is typically used, whereby we have our sample chamber here, and we have our GPMVs, which are not labeled, sitting on the bottom. And then we add fluorescent synuclein to this mix. And then we have our laser parked over here. So we're still looking at diffusion through our focused laser over here. But what we're then doing is we're watching our loss of sample as it binds to the GPMVs, right? So as molecules begin to bind, then we have fewer molecules left for us to measure. Right, so I don't want to dwell on equations, but this is the equation people typically use to fit three-dimensional diffusion, which would be appropriate here. And all the data I showed you before had this diffusion time as the primary parameter that we were interested in fitting. But the other parameter that you get from this data is this 1 over n, or 1 over the number of molecules, right? And so what we can do is we can watch our number of molecules decrease as a function of time or concentration or whatever. And so this is our quantification here, where after incubating for 1 hour, we found that we had, in untreated cells, we had about 20 of our, and this is roughly nanomolar, 20 nanomolar synuclein left in solution. Whereas for those cells we treated with PNGase F, we had about 60 nanomolar left in solution. And just to orient you here, we started with 80 nanomolar. So that's what that red line is, right? So we can see that, roughly 75% of the protein is bound for the untreated GPMVs, whereas only about 25% is bound for the treated GPMVs, right? So clearly there's some sort of, at least the glycan, which is important for this type of binding, right? And so our hypothesis is that you have some sort of glycoprotein in SHSY5Y cells that is important for this interaction. And if the sugar group in particular is important, and that protein is lacking in hex cells, which is why we don't see it here. And so, the cell membrane, the GPMVs are really nice because they lack active uptake. And this will be my last few slides. I don't want to go too long. Sorry, Tim, I see it's getting a little long. But I just want to show you that we can use the same approach for looking at uptake in cells. And that's what we did here. So now we're not just looking at binding, but we're actually looking at active uptake. So by imaging, we can see in untreated cells, when we incubate them with alpha-synuclein, you see internalized all these little endocytotic puncta, which is the synuclein that's been internalized. This is after about 6 hours, so it hasn't gone too far in the lysosomal pathway, but that's kind of where it's headed. And for treated cells, we see much less of this protein being internalized, right? So removal of the glycans also, as we might assume from the fact that it impacts binding to GPMVs, it also impacts internalization. And again, we're using FCS to quantify this. So rather than just with GPMVs, we now have cells and cell media. We park the laser up here. And now as a function of time, what she's doing is she's monitoring the loss of protein from our solution, just like we did with the GPMVs. And by doing this as a function of time, you can see we get really nice dynamic information about this, right? So for the untreated cells that I'm showing you here in gray, you can see that we've basically by about 5 hours, all the protein is internalized, whereas you do get some internalization, for the treated cells, but it's much, much slower, right? So we can actually not only quantify how much is internalized, but then the kinetics of that process. And just to sort of, bring this back a little bit to the biology we saw with the GPMVs, with hex cells, you don't see any of these puncta, right? They really don't get internalized, which maybe is what you would expect. If it can't bind to the cell membrane, it's not going to get internalized by the cell. And just the primary neurons behave more or less like the SHSY fibrous cells. So that suggests to us there's some neuronal partner also found in these neuroblastoma derived cells that is important for this interaction. So I'm going to stop there, sort of to, with that bit to allow time for questions if there's questions. And I'll just go back to that real quick. And I guess I lied to you. Maybe it was 110 minutes. And then I put my people back up so I can thank you for your attention, and then thank them for the work they've done. And then I'm happy to take questions.
Absolutely brilliant. Let's indeed give Liz a round of applause. So thank you ever so much for that, Liz. And there's been a bit of chat in the chat. No specific questions, and people saying some great talks. Does anyone have any questions for Liz? I'll kick off with one, if not. So I've always been fascinated by this, the distribution question that you show, Liz. And so when you take these measurements, does that mean you're taking multiple sort of 10-second measurements of the same sample? Or could you just give us a little bit of because if you just, are you taking one long 1 hour measurement and chopping it up into little bits and then analyzing each individual section differently? How are these measurements distinct, and how are they different?
Yeah, that's a great question, Tim, because I think they're basically people that sort of have their strong feelings about doing it one way or the other. So we are of the, multiple short measurements school, right? So, in each of these cases, you take somewhere in the order of, let's say, 50 curves, especially when we're looking at distributions and you need a lot more data, right? Somewhere between 10 and 50 curves of 10 seconds each. And honestly, though, we do that because that's how our correlator works, right? So it's a hardware correlator. It's actually quite ancient. When it dies, we're going to be in big trouble. But
no, you're not. You're going to buy one of ours. It's fine.
You won't be in big trouble. And so that's how ours is set up. And so it's easier both from a software and hardware perspective to do these multiple short measurements. It also, it's from a, in many cases with these proteins, like, you'll start to lose signal at some point, right? And so it's easy then to see when your curves start to go down, and you can say, okay, we're going to only take the first half of our data set as opposed to the whole data set, because clearly we're getting some sticking or something weird going on in the second half. But I think equivalent, it should be equivalent to one of these 1 hour measurements where you chop things up as well. It's just that because of our software and hardware, we do it the other way.
That's brilliant. And so is that a fresh sample? Do you re-prepare everything on and all of the rest of it into a new well as well? Or
Yes, and that's because tubulin is a really, really painful protein to work with. So tubulin is one of these proteins that you do purify from animal sources. So it's purified from a freshly slaughtered animal brain. And it, you can use it maybe for an hour, at least, according to my lab members, that before you start to get it just basically aggregating on itself and falling apart. So for us, then again, it's always a fresh sample so that we can make sure our tubulin is behaving properly.
I'm pretty sure I remember a whole pig turning up at Yale at one point.
Yes. At some point, the slaughterhouse made us buy the whole pig instead of just the brain. And so I think my lab had a clever spreadsheet to distribute pig parts.
Distribute the bacon. Yeah, no,
exactly right.
Brilliant. There's a question in the chat. Abigail, if you'd like to unmute yourself and turn on your camera if you want to. Don't. If you just go ahead. The floor is yours, Abigail.
Hi Liz, thanks. That was a great talk. I was just wondering, since you kind of looked at the role of glycans in the binding of alpha-synuclein, if you'd considered perhaps the role of the perineuronal net, and if you'd thought about kind of trying to replicate that system and how that might affect the alpha-synuclein binding.
No, but that's a really great question. Yeah, we haven't done that at all. We've been trying to, I guess where we've been putting our energy recently is to try to move away from rodent systems to work in iPSs, with the idea that at least then we'll have human-based cells, and that's probably a little bit better for in terms of sort of compatibility between the different proteins. But I had not thought about the perinuclear net, and that's a really good question because but no, we haven't, and that's a great point. We might want to think about that. I don't know if there are good model systems for that.
I think if you're familiar with Jessica Kwok, you might find she works with HEC-PNN cells, so it might be useful for you, but I'm not sure. But thanks for answering the question anyway.
Brilliant. And one from Rodrigo. Do you want to unmute and ask your 2 quick questions he promises in the chat anyway.
Yes, thank you. Thank you, Liz. One quick technical question, because I'm not too familiar with FCS. When you have the diffusion time in the y-axis with the different constructs that you tested, what is where does the dispersion in the x-axis comes from?
In the x-axis, that's just plotting, right? We just want to be able, if we plotted all the data at a single point, then you would not be able to really see the distribution. So the x-axis,
it's just plotting.
Yes,
okay,
exactly.
Wonderful. Thank you. And the other question is, maybe I missed this, but if the N-terminal domains inhibit polymerization, how is this? Does this affect this in biology in general? Like, is there another set of signaling events that moves away, or
we my student who did this work, she also did a really nice single molecule fret, which I didn't talk about at all. Let's see if we can get to this right, where basically the model she came up with sort of based on this data and then the N-terminal inhibition and then her single molecule fret, where she's able to show that this N-terminal domain basically, in the absence of tubulin, is making interactions with the proline-rich region and MTBR, which, sort of makes sense in terms of, and that once it binds tubulin, you basically lose that interaction. So this becomes much more extended and no longer makes those interactions. So it's probably charge shielding to some extent, because the proline-rich region and the microtubule binding region are very positive. This N-terminus is very negative. Tubulin is very negative. So as you sort of begin to drive that interaction here, you lose that electrostatic attraction, and this begins to sort of open up. So she proposed it as a sort of gating mechanism, and that, it's making this interaction over here, and you lose the ability to shield or this area as you begin to bind tubulin. And so that's the cartoon we have in our mind. And it's interesting because you have a fair number of modifications in the N-terminus, as well as disease-linked mutations in the N-terminus. And it was never really clear why mutations down here should necessarily impact aggregation. But one way they could do this is by altering sort of this, ensemble of the way it interacts with the proline-rich region or the MTBR in the absence of tubulin. And so that's something we're looking into now.
All right, thank you so much.
Awesome. Any final questions for Liz? In which case, it just remains for me to say thank you again, Liz. That was absolutely brilliant. Really great to see the work. Lovely to connect. I look forward to seeing you when we're next over the, well, not next. We're actually heading over for a couple of installs in the next few weeks at a couple of different places on the other side of the pond. But yeah, when we come to UPenn for biophysics, that will be fantastic. So thank you everybody for joining us. Look out again on Twitter and LinkedIn and in the emails for the next exciting seminar. We'll have a break over the summer and flood your inboxes again come September. But Liz, thank you again. Fabulous talk, lovely stuff. And
thanks everybody.
Cheers. Thanks everyone. Bye for now.
Bye.